Hello, everybody. I'll be your host today. And before starting the talk, I would ask you just to check if there are no technical glitches. And so in the participant box, you can raise your hand. If you could raise your hand shortly, just to see that you can ask questions. Thanks, perfect. That's perfect. Thank you very much. Go and lower the hands. Perfect. So, so this talk will be streamed, and so everybody can, at the end or in between, depending on how time goes, ask questions directly, either on the chat box or via personally. If you raise your hands, I can bring you in and you can ask your question directly. And so depending on time permits, I will try to handle all the question and answers, the questions so that Gasper can answer them appropriately. Let's hope that there are not too many or that I can handle them correctly. So it's my pleasure to introduce Gasper Tkacik today. He's originally from Ljubljana, where he studied math and physics, moved on for his PhD to Princeton, with where his advisors were Will Bialik and Curtis Callan. After that period at Princeton, he moved further to UPenn to work with Vijay Balasubramani and Phil Nelson. And since 2011, he is one of the, we could say, founding fathers of IST. Shortly after this institute was founded, he started working here and has been here in the last almost 10 years. I mean, Gasper's research interests are pretty wide and ranging from theoretical framework of gene regulation, collective behavior, evolution, to biophysics, computational theoretical neuroscience. Thus, if you look at his publication list, you see an exquisite diversity of collaborations and interesting projects. And today, Gasper will talk about the general problem in biology, how to approach higher dimensional problems with a normative perspective. And his title, as you all know, is Spanning the Arc Between Optimality Theories and Data. And with that said, Gasper, thanks so much for giving this talk, and the online stage is yours.
All right, thanks, Max. You can hear me, I hope. Yes? Yes. All right, perfect. So thanks everyone for tuning in. As Max said, this will be a talk about trying to tie together some loose ends between statistical inference and doing optimization or optimality theories. I'll explain how that goes. But first, I'd like to acknowledge the contributions from people who have worked on this project. That's Thomas Sokolowski, who was a postdoc here, but in particular, I would like to highlight the work of excellent PhD student Misha Hledik and Viktor Mlynarski, who is an IST fellow here at IST. All right, so let's get started. The outline of the talk is as follows. I'll spend a few words, or actually I'll spend quite a lot of time trying to introduce this notion of optimization theory, of normative theory, as we call it, and contrast and compare this with the inference or fitting of statistical models. Then I guess the sort of theoretical meat of the story is here, point number 2, which is introduction of a Bayesian framework in which these two approaches to problems can be merged together in a consistent fashion, in a statistically consistent fashion. And then depending on how much time we have, I would like to illustrate sort of this application of statistical ideas on 3 examples. So I'm sure I can cover example 1 and 2. One is really inference and optimality in a very simple toy model, LN toy model. And example number 2 is an actual application of these ideas to actual data, to receptive fields of neurons in V1. And then I think I'll pause for questions there. But if time permits, I would then like to bring up a very different example that's not even from the world of neuroscience, but the theoretical ideas are much the same. And that is about biochemical networks. All right. So, you know, to set the stage, I would say, at least from my personal perspective, optimality theories have really played a central role in biology. They have been central in how we deal with biological complexity. And maybe the clearest example of this is, of course, the theory of evolution itself. Here is just a schematic diagram of how various individuals, here these dots, the colored dots in some genotype space, through the action of mutation and reproduction are climbing towards the fitness peaks. That is where their reproductive success is the highest. And so the whole theoretical framework of evolution is actually based on the notion of optimization of fitness. Of course, with sort of lots of complexity I'm skipping. Now, this overall idea that fitness is being optimized throughout evolution then found its manifestations in other types of systems. For instance, here is a famous paper in the metabolic network of E. coli. So this is a set of metabolic reactions or subset of metabolic reactions in a bacterium that consumes nutrients in order to build more of itself. And this theory, and one can build a theory from this complexity here by postulating that various fluxes of metabolites through these chemical reactions that we know and we can write down their stoichiometry, that chemical fluxes through these reactions have been set by evolution so as to maximize the growth rate. And that generates a very predictive theory that actually predicts what these fluxes should be. One can then go and measure those and ask, is the growth of bacteria really consistent with this optimality? I.e., subject to metabolic constraints, do bugs grow as fast as they can, basically? So here you see a very different example. It's an application of optimality theory to a transportation network. So what you're seeing here is a particular type of an organism that is sort of building, this is a kind of a nutrient in the shape of a bay of Tokyo. And these lines here represent the organism that has created sort of a tubing in such a way that it is extracting nutrients from this part, and this part here does not contain any nutrient, in a most efficient way, that is with the least tubing possible. So basically the organism, while it grows, is solving this transportation optimization problem. And one can ask, is it solving it optimally? Is the solution similar to what you would find in the computer if you were to optimize the same transportation problem? So examples abound. I will mention just one more example, because this is something that we have been doing for essentially 20 years now, since my PhD, which is the optimization of information flow in biochemical networks. So here the example is of certain types of chemicals in your cell called transcription factors. These are proteins that regulate expression of genes from the DNA. So their concentrations, as they change, they bind to regulatory regions on the DNA and turn on or off genes. So the gene expression goes up and down. And one can ask whether the information flow from the concentration of transcription factors to the gene expression levels is somehow optimized by the action of evolution. And this is actually sort of setting the stage for this final example, if I can come to it. Now, pursuing this last thought that not only might growth rate be optimized or the flux of, the flux of matter through bioreaction networks, but that also the flow of information might be optimized. So those ideas actually trace back to the very beginnings of information theory by Shannon. So let me just say a few words about that. So Shannon's information theory was a theory that was built actually to solve an engineering problem. That is, to solve a problem of how you can transmit most easily, with least errors, most efficiently, information from some source. Maybe that is a person speaking in a telephone line, to some receiver, which is perhaps somebody listening in at the other end of the telephone line, and that could be an automatic system as well. And that theory is fundamental to how quickly we can transmit the information today in mobile phones and so on. So the central object of this theory is this notion of a channel. It's a mathematical concept. The channel is this device that transmits the information. So the information is created by a source. It could be me speaking, or it could be a page of English text. So that's X. And now this X has to be fed through the channel. At the output of the channel, we get Y. Now the channel is imperfect. So if it were perfect, that for X that you put in, X comes out without any error. But because the channel might be, as said, a telephone line or a transmission tower with interference, with a limited power and so on, what comes out at the receiver end is not X, it's Y. And now at the receiver end, what you want to do is you want to recover without errors or as few errors as possible what was being sent, right? So the central conceptual notion of this theory is this notion of a channel. We have input signals that go in, the channel has cost, has noise and so on, and at the receiver end, we try to reconstruct the signal. Now, Shannon then defined a mathematical quantity, which is called the mutual information between what is being sent and what is being received, X and Y. It's a number. That's the thing that you know that's measured in bits. There is an equation which I will not focus on, but this mutual information basically tells us, it's a sort of in a way, it's a unique measure of how well information can be sent without errors through this noisy channel. And the interesting thing is that Shannon already realized that this notion is so abstract that it probably doesn't apply just to engineered systems. And actually in the book by Weaver, you can read, Shannon and Weaver, you can read that we can use a very broad definition of communication, one which would include the procedure by means of which one mechanism, whatever that might be, can affect another mechanism. And sure enough, this sort of breadth of information theory was evident because it started being applied to problems in biology. So 1948 was the theory. And then here is a brief timeline of when people started thinking about it in context of DNA and so on. And what I would like to highlight is this guy here, Horace Barlow, 1961, that proposed what is known now as the efficient coding hypothesis. So the efficient coding hypothesis is the following application of information theory to biological information processing systems. It says the following thing. It hypothesizes that evolution has optimized the flow of information, so has maximized the flow of information as defined by Shannon through, say, our sensory systems, let's say in our eye, subject to hard biophysical constraint. For instance, that neurons in this system cannot emit more spikes than let's say 100 hertz, okay? So that's a first principle, is an ab initio, that's a normative theory, it's an optimization theory, okay? So here, this paradigm of Shannon, of short source, channel, and the receiver, can be applied to biological problem. For instance, the problem of how light signals that are incoming to the retina, right, input X, can be encoded by neurons into spike trains Y, and these spike trains are subject to various constraints, maximum rate of firing, maybe some noise constraints. And then these signals are being sent from the retina, say, to the brain, which then needs to recover signals of relevance. It could reconstruct the image, maybe it needs to estimate something about the external world. Now, the same thinking is applicable also to very different systems, as I was alluding to. So for instance, X could be also a concentration of a chemical in the environment, a ligand, or it could be a concentration of a transcription factor in the cell. And then the channel that's receiving this concentration could be, say, a gene regulatory element, maybe a promoter or enhancer, which as the output yields a particular level of gene expression. It could be also, the source could be also a concentration of a chemoattractant that a E. coli bacterium is responding to through its channel, which is a signaling network in the cell. And the output of the system are movement decisions, where to swim, towards the chemoattractant or away. And so in all these examples here, information theory can provide a way to understand these biological processes within one abstract framework, which is of these communication channels, which has a well-defined mathematical formalism, and also using one universal currency, which is a currency of bits. Now the question is that approach productive? Does it predict something, okay? And so here is an example where such predictions were obtained, or applications of information theory were made in sensory neuroscience. I'll focus on perhaps the best known example, which is in early vision, where the inputs are set will be light signals and the encoding will be by neurons, say here in the retina. All right, so this is a very important point. So information theory has been used in this context in two very different ways. So the first way is it has been used as a means to quantify how much information flows from the images through the retina to the brain. This was done by actually measuring real spike trains from retinal neurons, and then estimating how many bits per spike or bits per second, as you know, as a function of the firing rate, various neurons are transmitting to the center of their brain. So here, from this review by Peter Sterling and Balasubramanian, every point is a neuron, and so you see that these neurons typically convey 2-3 bits per spike, bit going down with the spiking rate. And if you now extrapolate to the whole guinea pig retina, GP, the total budget of information flow through the optical nerve, you will estimate from the data, without any theory, that the information flow through the optic nerve is about 1 megabit per second in the guinea pig. Now, you could use now information theory, exact same theory, for a very different goal, which is to ask about optimization, to ask how each one of these cells, of retinal ganglion cells, should actually behave, how it should process natural images. And the showcase result of this thinking is this efficient coding result, which was to say, if each neuron is to act on the stimulus as a linear filter, and the output of this linear filter predicts the firing rate, what should the linear filter be? If you now say that that linear filter should maximize the mutual information that the neuron conveys from stimulus to spikes, and if you assume that stimuli are real natural scenes that you can see outside and record by the camera, and you say that neuron has a maximal firing rate, then you can derive what this receptive field shape should be, and it comes out that it should be a center surround that you all well know. So sum up pixels in the center, subtract away the value of the light intensity in the surround, and that predicts the firing rate. So that's a priori theoretical derivation from a normative principle, and you can then compare it with real measured receptive fields to see whether this theory has correctly predicted what neurons should do. Okay, and indeed, you know, this prediction of center surround receptive field was a hallmark of efficient coding, but it's not the same or not the only prediction. You know, there is a list of predictions, which I'll show briefly, but the point here is that there are two uses of information theory. One is to quantify information transmission from data, from actual data. The other one is to predict what this processing in neurons should be from first principles, and which you can then compare to data. Okay, so the prediction of the receptive field shape is by far not the only one. Just in the retina, there is a whole list of successful predictions that are listed here in various papers, going from adaptation to the ratio of various color receptors and the ratio of on and off cells and so on. There are, of course, things also in the retina that have not yet been explained in the context of this efficient coding, just to you know, full disclosure, of course. Now, the main challenges of this second line of thought, where we optimize and then compare to the data, and that I want to focus on, are as follows. So the first one is, I've shown you the prediction of the center surround receptive field, and I've shown you the measured receptive field, and I said, look, they look alike. But I asked, I want to be slightly more rigorous. Can I derive from this a real rigorous statistical hypothesis test from optimality? So here, just to make this very concrete, here is a picture from a beautiful paper by Jan Karplin and Nero Simoncelli. What they did is they built a computational model of the retina that they optimized. So they optimized receptive fields of these cells, and so they optimized temporal receptive fields and non-linearities that are shown here. So this is a theoretical prediction of what cells in the retina should do. There's many more cells in the retina, like a full mosaic. They optimize all of that. So it's a big optimization problem. They get these nice predictions, center surround receptive fields, and this kind of non-linearities and sort of biphasic temporal receptive fields. And so they plot this prediction, and they plot a real receptive field from the retina with their real inferred non-linearities. And so the question, and you know, they show both and say, see, this is an example of a successful prediction. But what I want to know is how to make that comparison of theory and data really rigorous. Is that really consistent or not? Okay, so is the retina optimal or not? The second thing is, maybe I need to relax this criterion and ask not only is the retina optimal or not, but derive a measure, continuum measure of whether the retina is close to optimality. Maybe it's too much to ask whether it's really optimal or not. Maybe it has not been fully optimized by the evolutionary process. Maybe our idea of what optimal means is not exactly correct. So perhaps this yes or no test for optimality needs to be relaxed to define a notion of closeness to optimality. Is it 90% optimal? Is it 95% optimal, or so on? So this I would also like to do in our framework. Point number 3 is more difficult to explain, but I'll try. Oftentimes our optimization theories do not generate unique predictions. Okay, so for instance, I said a prediction of an efficient coding is a centers around receptive field, all right? But that prediction might depend on the constraint. You remember, the prediction is under a constraint that neuron can only fire so and so many spikes per second. So that I need to put into the theory. I said, like, if it spikes at most 100 Hertz, what's the best receptive field? But sometimes I might not know this constraint. So maybe here in the case of maximum firing rate, I have a good idea, but there are theories where I know that there needs to be some sort of a constraint, say on the energy expenditure of the neuron, but how much exactly it is, I don't know, okay? So how do I derive predictions then? They depend on this constraint, which I do not know, so somehow I should fit the constraint to data simultaneously while I optimize the other parameters, okay? So I'll try to make that notion clearer in a bit. And then the last point I want to introduce is when I infer models from data and I have some idea that the system might be optimal, can this idea of optimality actually help me infer models from data? And if so, how? Okay, so these are the challenges. These challenges become really limiting for complex models, I'll claim. And so the goal here is to derive a mathematical framework in which these questions can be jointly addressed. Okay, so to go there, let me now contrast even more optimization theories here and statistical inference. So top-down or a normative optimization theory has the following goal. I want to find a model that optimizes some notion of biological function. This notion of biological function is given by some utility function that needs to be maximized. So I don't care about, in this approach, I don't care about the data. I just care about the utility function. Maybe information transmission needs to be optimized with the context of some model, probabilistic model of neural responses X, given parameters theta. So given the utility function and the model, my optimization will predict the best value of parameters that maximizes the utility function. So what is the receptive field that maximizes information transmission? So this is what optimization theory does. Now, statistical models, statistical inference does the following. We are looking for the best model that describes actual data. So here we start with data. Okay, so this is data driven, but it's agnostic about function, right? So typically what I do is I postulate a model, P of neural responses given parameters. I'm given some data, and then inference, which is just another word for fitting, consists of taking the data and using it to find the best value of parameters, say by maximizing the log likelihood of this model, right? So here utility predicts parameters, here data by fitting constrains the parameters, right? And so interestingly, these two approaches have traditionally been used very independently in parallel without too much intersection, and we ask whether they can be productively mathematically combined, right? And so here again, just to reiterate, here is our red normative theory, right? It's maximizing utility function. It's looking for optimal parameters, theta star, that maximize utility. Here is our statistical inference in blue. It takes real data and it's making the inference. And both of these, both the normative theory and inference, are making statements about parameters, in this case, two of them, theta 1 and theta 2 of the model, right? So normative theory says optimal parameters maybe sit here, that maximizes the function, and inference says, well, you know, if I infer from data, maybe they're here, right, as well. So so what we are claiming is that there exist not just the two extremes, which is the extreme red, where we don't use any data and we make ab initio predictions, and not only the extreme blue, which says we only care about data and we don't care about utility, we just use data to constrain parameters, but there is a whole range of interpolating regimes where we use a little bit of data to regularize theoretical predictions, or we use some notion of optimization to regularize inference, and of course there is the two limits that we already know. So how do you construct this continuum, right? And the main idea for this theoretical is, for this approach, is that we can encode this normative theory this utility function in a prior, in what we call optimization prior that I'll introduce. And once we have a prior and the data, we can consistently combine them using the Bayesian framework. Okay, so here is how it works. So if I'm given some utility function u, let's say information transmission from parameters, given parameters theta, I can make a distribution out of it, which is called the maximum entropy distribution. That's a prior over parameters. That is what I believe with absence of any data, with this beta parameter here. So this distribution is as random as possible while achieving a certain average utility. Okay, when I take beta to be very large, infinity, so to say, then parameters that, the distribution of parameters is the single point in the parameter space that maximizes the utility. That would be our normative theory, right? The single point. Okay, but for beta being finite, I have basically a spread over the parameter space that likes high utility parameter sets. Okay, and then of course I have the usual likelihood term. So the likelihood of the data given the parameters, which I can compute, which is the usual fitting term that I can compute using my measured data. And then the Bayes rule tells me I can combine the two to form a posterior over the parameters by taking, of course, the fitting term, the likelihood, and this optimization prior here, and combine both to get the posterior over parameters that depends on data, on my choice of utility function that I believe the system is optimized for, and this parameter beta here. Okay, and so now there are two regimes depending on this beta. Okay, so I can take a very large, strong beta. This emphasizes the optimization term, and if I take the beta to infinity, I'll completely ignore the data term, and I'll just predict that the optimal of the utility function. That's my normative theory for very high beta. If I take a very weak beta, then this posterior is dominated by data, right? So it's whatever data tells me, okay? And then the solutions of the model are, of course, determined by the model and the data, and this thing here is ignored. But of course, beta now interpolates between these two extremes. And I would say this is a very different use of prior. So here, the prior contains the whole theory. That's a very different use than if you're doing statistical inference, and the typical use of prior is just to do weak regularization, to try to keep parameters in zero and so on. This is highly structured use of priors, as I will show in the example. All right, so let me give you a first example of a toy LN model neuron. So we are thinking of a neuron that's receiving this simple one-dimensional stimulus. So these are its values, the stimulus values. The stimulus is passed through a sigmoid nonlinearity that has two parameters. So our model has only these two parameters, which is the position of the nonlinearity x0 and the slope, which is called k. And then the output of this nonlinearity determines the probability of making a spike, which is R, 1 if the neuron spikes and 0 if it doesn't spike. Now, again, the standard likelihood inference will say, I'm given some data points. So here are values of stimulus x. And whenever you see a white dot, the neuron did not spike. And whenever you see the black dot, the neuron actually spiked. Given such data, I can construct my likelihood for the two parameters, x0 and k. High likelihood is in blue. And the more data I have, the more this will be peaked at this particular value of x0 and k, which is the best fit for this model neuron. Okay, so that would be the standard likelihood inference. Now, I can think of the optimization theory. So then I need to say what kind of utility I think this neuron is maximizing. And I'll, in the context of our efficient coding, information maximization, I'll define that utility as being the information that this neuron encodes about the stimulus. So then I have to postulate that the stimulus is drawn from some distribution. In this case, we played around with the three-peaked distribution of stimuli. Let's say some stimuli correspond to stimuli emitted by a predator, some by prey, and some by a conspecific, for instance. Right now, this means I can define the information that this neuron encodes in its spiking mutual information, given the stimulus label, right? So this, or this. And that information will depend on where I put the neuron's nonlinearity and slope. So if I put the neuron's sigmoid in between these two peaks, right? So then whenever there is a predator, the neuron will not spike, but if it's prey plus conspecific, it will make a spike. That corresponds to a pretty good solution. That's a solution at, say, low x0 and high k. So low x0 and high k, so it's here. So if I put it here, I get almost sort of a bit of mutual information between the stimulus and the spiking. But you see that's degenerate. So I can also get a bit if I put a sigmoid, for instance, to go like that, or I could put an inverted sigmoid like that, or I could put an inverted sigmoid like that. So there's 4 very good solutions that separate 2 of the peaks from the third one. And then, and these are these dark red things. And then there is lots of bad choices, which are these not dark things, right? Where I put the sigmoid in the middle of this Gaussian, for instance, here. Okay, so this is my utility function. It's the mutual information as in efficient coding. And then from it, I can construct, so from this U, from this mutual information, I can construct this priors of a parameter space. If beta equals 0, that means the prior is completely uniform. I don't favor good information solutions or bad information solutions. All parameters values are equally probable, so I get a flat distribution over the parameter space. But the higher I make this optimization parameter, the more my parameters are localized to these spots where information is very high. So these spots here, right? And so as a function of this optimization parameter beta, I achieve utility plotted here from 0.4 bits up to 1 bit. And so the higher the beta, the more I go along this curve toward higher utility, and the smaller is the entropy of this prior distribution, which means the entropy just measures how localized this distribution is. So very high entropy here, uniform distribution, very low entropy here because the distribution is localized to these 4 spots. Okay, so I get this curve. Okay, so now let me show you a first application of this framework where you combine the prior and the data in the sense of test for optimality. So if I were given some actual spiking data from such a neuron, and if I assume that it's trying to optimize information, could I accept or reject the hypothesis that that neuron is optimized for information transmission? Okay, so again, we have the stimulus, we go through the nonlinearity with the 2 parameters, and the neuron spikes or it doesn't spike. Here is my utility function as a function of these 2 parameters, slope of the nonlinearity and its position. And I'll consider 3 examples. So a neuron that sits here, obviously very suboptimal, okay? A neuron that sits here, which is kind of not completely optimal, it's like half optimal, right? So it's dark red, but not the darkest red. And then 3 sits exactly on the peak of the utility function, so that's really optimized, okay? So imagine I see from each of these 3 neurons, I see a few spikes, several tens of spikes, okay? If I do that, I can localize the parameter values for neuron number 1 into these regions somewhere. Remember, it's a finite number of spikes. So I have so there is sort of the likelihood is not a single dot. It's this shape here. For the neuron number 2, I localize the parameters here, and for neuron number 3, I localize them in this kind of peak of high utility. Now, why is the question of whether any of these neurons is optimal or not easy? Well, for 2 reasons. So the first one, if you look at the shape of the utility, there is no clear separation between what's really optimal and what's not. It's a continuum of values. You know, this is bad, this is very good, but there is all stuff in between. So it's not black and white, okay? And second, not only that utility is not binary, also exactly, given that I have finite data, also exactly where my neurons parameters are, it's smeared because I don't have an infinite amount of data. So how do I now test, is the neuron optimal or not? Okay, so the proposal that we came up with is to define what is statistically known as the marginal likelihood of this beta value, the beta that goes into the optimality prior, given our data. So that is essentially an integral over the parameter space of the product between the likelihood and the utility. So that's a single number that shows how likely it is, given that we have observed some responses, that this optimization parameter beta takes on a certain value. Okay, so let me show. So for this, so this is easily computable in this case. For the first example neuron, here is this marginal likelihood as a function of beta. So high beta means very optimal, low beta means not optimal, beta 0 means not optimal at all, kind of random in some sense. Okay, and for the first example, you see that this marginal likelihood peaks at beta equal 0. For the second case, this neuron here, which is kind of not at the peak of optimality, but at the flank, there is a non-zero value of beta, but it's kind of small, 3.7 in the marginal likelihood. And for this case, which we know sits exactly on the optimum, I see that the marginal likelihood is maximal at this highest value of beta that I tried. Okay, so it doesn't saturate. And then if I want to make a rigorous statistical test, I can define a test statistic, which is the likelihood ratio, lambda, that the system is at this beta hat value compared to the fact that it's divided by the fact that it's a beta equals 0, which is sort of a uniform, non-optimized system. Right, so that's now a test statistic. And for this test statistic, I can actually compute the null distribution. So here is the distribution, null distribution under the hypothesis of no optimization. So this is this line here, the blue line. Under the hypothesis of no optimization, this is what the distribution of this lambda would be. And if I ask now where my 3 neurons sit, 1, 2, and 3, they sit here. The neuron 0, the neuron 1 is at this lambda equals 0, neuron 2 is here, and neuron 3 is here. So if I now draw a significance threshold in my null distribution, I can see that only this neuron number 3 passes that significance cut. It's in the tail of the null distribution, while neurons number 1 and 2 would be consistent with random, given my, I think in this case it's like a 5% significance threshold. Okay, so this means I can derive, in this simple toy example, rigorous optimality hypothesis testing under a hypothetical utility, which is in this case maximizing information transmission. All right. Now this can easily be extended. So this was for a single neuron. Suppose now we observe n neurons, measure n neurons, let's say n neurons from the retina, n neurons from the visual cortex. Can we ask how optimal they are as a population? I'll just briefly say that this can be done by turning the crank of hierarchical Bayesian models. So I can imagine that I have n neurons, each of them with their own parameters, theta 1 to theta n. Again, these thetas, let's say in our case, are the position and slope of the nonlinearity. And I imagine they all share the same value of the optimization, right? They're all realizations of some system at a certain level of optimality given by this number beta that we don't know and we would like to determine. And from each of these neurons, we observe some experimental data, some patterns of spikes, say, right? So again, here are 3 toy model examples. Here I have a very high bet. So here I'll generate my fake neuron, my synthetic neuron data by drawing from a highly optimized distribution, beta equal 12. So red dots are realizations of my neurons, and you see they sample from places of high utility, from these 4 lobes, okay? So here, these are low optimal, beta equal 4. They kind of predominantly are in the region of high utility, but some are off. And here, these are non-optimal, I just sample from the middle, from some Gaussian distribution, right? Which does not sit where high utility solutions are. And I imagine that from all of these red dots, I would generate spike trains. My experimental data would be spike trains from these kinds of neurons. And now by turning the crank of the Bayesian framework, I could take this data and ask about the posterior distribution over optimality parameters for the 3 cases. And so for the first case, these highly optimal neurons, I would find that the posterior over beta is sharply peaked around 12, which is precisely where of course, I mean, it's a synthetic model, so it has to come out right. This is of course where I generated data from. And for this medium optimization, I would get beta peaked at 3.75, very close to 4 that generated the data. And for these Gaussian things that were not drawn from any of the maximum entropy distribution, but just from some Gaussian distribution in the middle, they correspond to beta equals 0.2, so very, very low optimality, okay? Now, I can ask the question, like, how do I, like if these were actual data and I infer that beta is 12, how optimal is that? I have no scale for beta. So to solve that problem, what you can do is you can transform the beta values into the fractions of my utility as a fraction of the maximum that is reachable, right? So the best, as you know, as you remember, in this theory, the best neuron that maximizes information transmission gives me a utility of 1 bit. So this is here. And neurons that have a beta of 12.03, you can compute because there is a unique mapping between beta and the utility that I've shown you on 2 slides back. you can compute that these neurons have a utility of 0.93. So now this utility has a scale, right? It's on a scale of 0 to 1, and the optimal solutions, these guys, or almost optimal solutions, these guys reach a utility of 0.93 out of 1. These guys here have a utility of 0.8 out of 1. Now a uniform sampling over parameters would be here, and these Gaussian guys are actually below the uniform sampling because they're really concentrated in the bad region. They're not even uniform across the whole space. They really sit here where information transmission is very low. Okay. So the point is that this approach is not a yes-no hypothesis test. This approach is an approach to infer the degree of optimality beta, and that can be a continuous number going from really not optimized through semi-optimized to very optimal. Right? In biology, it makes sense to think of degrees of optimality, perhaps better than a yes-no hypothesis test. All right. I should also say that if you think a bit, given, depending on how exposed you are to statistical inference, this approach also properly controls for the value of the parameter space, right? If the parameter space was much larger, so if this box was much larger, and the peaks were very tiny, right? This transformation from beta to utility tells you also something about how unusual it is that you are so close to the optimum, given that all the parameter space is that big or slightly smaller or slightly bigger. All right, so the last application is an application where, or is an example where optimality theory actually helps us fit the models, right? So this is now a bit more realistic. So imagine we have stimuli which are not one-dimensional now, are full image patches, natural image patches. This stimuli will be, like in the case of retina or V1 neurons, convolved by a filter. And then that goes through a nonlinearity to determine Poisson spiking. So what we will do now is we'll do the following. We will assume in our synthetic world, in our model world, that the neurons that we are working on are neurons predicted by some optimization principle, in this case, sparse coding. That is a very prevalent view of what happens in V1. That is that neurons are use linear filters that maximize the sparsity of neural responses. So either the neurons are very quiet most of the time, but when they spike, they emit rates that are far higher than what would be predicted if this was a Gaussian distribution of responses. So we'll generate ourselves sort of a bank of synthetic neurons, of sparse coding neurons for natural scenes. Some are optimized for the sparsity utility and some are optimized for some other type of utility, which is kind of sparse, but not exactly fully sparse, okay? We'll simulate the spikes, we'll pretend these are the real spikes, and then we'll ask, how can we infer receptive fields from those spikes? And can optimality theory help us do that? Okay, so here are two example neurons whose ground truth receptive fields, because I made them, are shown in these two panels. And now, if I were to do the usual normal inference of receptive fields, what would I do? I would take the spikes of these neurons and I would look for the receptive fields of these two neurons shown here by maximizing the log likelihood of the neuron's responses given the stimuli, right? That in our framework corresponds to not using any prior at all, which is beta equals 0. And given 2000 stimulus response pairs, I would get these type of receptive fields that you can compare with the ground truth ones, and they are pretty good, quantified by this cross correlation between the inferred receptive field 0.77 and the ground truth receptive field. For this guy, the correlation is 0.83. Okay. But now, what if we do not the log likelihood, not the likelihood maximization, but we maximize the posterior using our sparse optimization prior? Okay, so what happens? I can now do maximum a posteriori inference of receptive fields by using priors with higher and higher beta. So I put more and more of optimization theory into my inference. And what you see is that when I use my optimality priors, I actually get a better prediction of the receptive field. So cross correlation goes from 0.77 to this one, 0.89. In this case, from 0.83 to something like 0.95. Now, if I crank beta even further, then suddenly the prior totally dominates the solution, the data is useless, and I just get solutions which are solutions of the optimization theory, completely different place receptive field, right? So these are in the regime where data is now completely ignored. But somewhere in between, there is a sweet spot where I use some of my optimization theory as a very good prior to drive and support the inference. So what this shows is that these optimality priors that come from theory are very highly structured. They act as powerful regularizers for inference because they capture the notion that the receptive fields need to be smooth, that they need to be localized, that they need to be band passed, that they need to be oriented. These are all the things that we feel V1 receptive fields should be. Now, when I use them for inference, I don't know which beta to use, but for that, I can just do classical cross-validation. Right? I pick the value of beta that maximizes the performance of the inferred filter on the withheld testing set. Okay, and what's even more, I'm not showing that here, but we actually did that analysis. Using these optimization priors helps inference even when the optimality theory is not exactly right. So even if I took the filters that I got in my toy example when I did not optimize for the say utility function, so long as they are smooth and localized and bandpass and so on, using this prior will actually help with some beta. I'll get better inference for that. Okay, so I can use these structured priors for inference even when I don't know exactly what the optimality theory is that the neurons are maximizing. Okay, so we are nearing the end. I'll just give this more concrete example. So we'll now depart the world of toy models, and we'll have time just for this example. Then I'll take a few questions. So these are now this is now an attempt to use all of these ideas, not on toy model scenarios and so on, but on real macaque V1 receptive fields that were published by Dario Ringach's group. So there is 200 in the data set that's published. There is 250 receptive fields of V1 cells, and a selection of which is shown here as a 32 by 32 pixel RFs. And what we'll now try on this on a single slide to go through the ideas that I've shown up to now on toy models. Okay. So first of all, we said we will consider two sort of optimality theories, both of which have been actually used for the cortical V1 neurons. One is the sparse coding utility function that I explained before. This is this sparse coding theory that predicts localized Gabor-like patches. And the other optimality theory that was also invoked was the so-called slow mode analysis, which says that the receptive fields of neurons should optimize, should be such that the outputs of these neurons are vary in time as slowly as possible. So these neurons are extracting predictive slow components of natural seeds. Okay. Now, if you just simply look at what the predictions of this theory would be, a sparse coding predicts a set, this oriented, localized, Gabor-like receptive fields. And the slowness utility predicts this sort of low, so long wavelength receptive fields, because that's what, what contains the slow components. So again, full disclosure, of course, we knew from the start, because many analyses have been done, that V1 receptive fields are typically viewed as being more consistent with sparse coding. And in particular, we focused on properties of single neurons, not of whole populations. Okay, but let's just carry through to see that all these this whole statistical framework is actually applicable on something that's real data and it's really high dimension. Okay, so the first thing we defined was an optimality test. Given a utility function, are the observed neurons consistent or not with that utility function? Okay, and so if we look for the sparse coding utility, so here what you see in red is a null distribution of utility functions, assuming no optimization. So that would be in red. This is what is predicted by our theory. And then the dots in the two colors here are the values of the utility for the actual 250 observed neurons. What you will see that for the sparse coding on this side, 204 of the neurons lie in the tail of the distribution, so in the top 5 percentile of the null distribution. And only the 46 neurons lie here sort of below the significance threshold for optimality. And if you actually look at what the typical neurons look like, for the green neurons, they're really these Gabor-like localized tiny ones. And the 46 ones that fall below the threshold, they're much closer to this long wavelength, smooth receptive fields, right? Having said that, this means that the majority of the neurons would be sort of accepted by sparse coding optimization theory. Whereas if you do the same analysis for slowness, what you would see is that most of the neurons would fail the statistical significance test. They would not be predicted to be optimal, right? 297 of them, and only 3 would actually pass some sort of a, not very high threshold here, right? So this would be yes, no hypothesis testing. Alternatively, you can do what we said. You can try to not do yes, no testing, but test the whole population and ask the population as a whole, using this hierarchical Bayesian approach, is it optimal or not? And thereby you can construct a posterior distribution over optimization values, over beta values, given the data. This is this purple color here with the mean at 3.4. And if you compare this with what you would get if you computationally optimize the same utility function, you get a distribution in red, which is just slightly higher mean at 4.5. And just for reference, here in gray is the anti-optimized neurons, so the neurons that maximize minus the sparseness, right? So this would be anti-sparse neurons, would be here. So this is for sparse coding. And so here you see that the real neurons in purple and optimal neurons in red show very similar distributions over beta. And for slowness, you don't see that. You see the real neurons take, this intermediate distribution here. This is the data V1, a posterior over from the data, and the optimized neurons for slowness would be actually much higher, would achieve much higher values of beta. Right? You could even ask whether the distribution of not just the mean utility, but also the distribution of utilities, given the data, which is this bright blue curve, matches what our optimization theory would predict, which is in this purple, which is a distribution of utilities given the inferred optimization. And you see that for sparse utility, you get, you should get the means, right? That's how we construct things, but you also get the width, right? So the diversity of the utility over individual neurons is consistent with the theory, the standard deviation of this distribution, whereas you don't see that for the slowness utility function, okay? So I will not show other things, but you can carry these type of analysis through on real data. All right, so tests of optimality of this sort are feasible on real data sets. And there are very interesting extensions that we have been working on. So for instance, here what we have done is a very pedestrian type of model selection between sparse utility and slowness utility. So we are essentially saying which optimization theory explains the V1 receptive field. And it's possible to do this much more rigorously using the tools of Bayesian model selection. It's also possible to consider utility functions that are parametric. So there is an unknown parameter in, say, the cost of spiking, and we want to test whether the neuron is consistent with some utility theory given a constraint on the cost of spiking, but we don't know the constraint. So we have to infer it at the same time as when we are testing. All right, so that's an interesting extension. All right, so I had a third example, but I would like to skip it and just jump straight to the conclusion slide, and then maybe take questions. So, even if you abstract away technical details and statistical details, the point I was trying to make is that it's possible to marry together in a Bayesian formalism inference, so fitting, and optimization. These are not two separated extremes. There is a smooth interpolation between top-down optimization theory and bottom-up inference from data. And these intermediate regimes where you mix a bit of optimization with a big bit of fitting are actually very pertinent and relevant for complex biological systems. One use is that you can rigorously test for optimality, yes, no. The second is that you can talk about distance from optimum, degree of optimality. One thing I have not shown is that this marriage of optimization and fitting, can set unconstrained parameters in the optimization theory, and also break the degeneracies in the optimization theory. In our toy model neuron, there were 4 peaks that are all equally good. As soon as you have some data, you can decide that biology is close to one of the 4, which one, okay? And in general, I think that this optimization, in biology, we deal with hugely parametric models, very complex models. I think the ideas of optimization can help us sort out that space, right? If we have some idea of what the function is, we can navigate that space better. We can focus on the parameter regimes which are consistent with high function, with good function. And this helps us in fitting and helps us building a real theory for biological systems. All right, and then the last bit I did not manage to show is the application of these ideas to a biochemical regulatory network. So hopefully this will come up soon as a publication. So let me stop here, and maybe we have a few more minutes for questions if there are any. Thank you.
Thank you very much, Gasper, for your nice talk. Please raise your hand if you have any questions. There's one in the Q&A box from JP. I don't know how to pronounce your last name, sorry. How do you account for noise correlations when fitting data are synthetic models?
All right, so I should say that all our data, at least in the toy model, were of neurons that are not full correlated populations. So they're conditionally independent neurons. We have not done a test of the actual coupled population. I think that's something I've wanted to do for a long time. There's actually a theoretical paper that we wrote in 2010 in PNAS, which asked about optimizing also noise correlations for information transmission, but we have not considered it yet in this setting.
So Naveen Pokala has a question. I mean, I ask it personally if you want. Yeah,
maybe.
Naveen?
Sure. Hi, can you hear? All right. Great talk. Thank you so much. Your mechanism, your algorithm sounds very similar in spirit to Kalman filters. And I was just wondering if you'd comment on some of the similarities or differences between your formalism of merging theory and data inference to the older Kalman tradition. Thank you.
Well, I would actually have to think in which case would you, as you say, reduce to Kalman filtering. I mean, in our formulation, you are very generic, right? So there is no assumptions about linearity. There is no assumptions of Gaussianity. So any probabilistic model can be merged together with an optimization prior that's max n prior. Now, of course, then in practice, you might want or have to do certain approximations to make this tractable, and in that case, perhaps that does reduce, although I'm not completely sure, in some regime to Kalman filtering. So I have not thought that one through. I think the formulation itself, right? I mean, up to the point where you embed an optimization theory, which you'd have to specify, right? If there is some link to be made with Kalman filtering. In our case, you're agnostic to what that optimization theory is, but once you specify it, you are turning the crank of Bayesian inference. So I think for Kalman filtering, you have to assume some Gaussianity and linearity, right?
Hey.
Thank you.
If Alberti calls.
Perfect. And we have one more question from Aditya, a fellow IST fellow here. How could you include a mechanistic model or mechanistic constraints?
Yeah. Okay. So I think for our group, that is actually one of the long-term goals, is to try to see how to go beyond the functional neural models and really talk about mechanistic models. Now, I think, in principle, this is not impossible, right? I mean, nothing in the approach constrains you to say choosing LM or a functional model. And the last example that I wanted to show you about the biochemical network is actually a mechanistic model. So there we just deal with, with sort of chemical reactions between transcription factors and binding sites and how they regulate gene expression. So I think the issue is not so much, can you do it in principle? In principle, yes, you can. The issue is simply technological. I mean, if, mechanistic models are usually parameterized by many, many parameters. And then some of the things I showed you in the toy model, which is, only has 2 parameters. So parameter space is 2D. If you need to integrate over it, that is trivial. You just sum over the grid. In the mechanistic model, so our biochemical reaction networks has about 50 parameters. And so there, it's just non-linearities are nasty, it's high dimensional. There is a lot more of compute that you need, and you need to use Monte Carlo tricks to get around the technical problems. But it's in principle, you can just put it in, since mechanistic models usually are recastable as probabilistic models, right? Because, biological systems have noise.
So if there are no more questions, you can raise your hand. Let's thank Gasper again for his nice talk. And thank you very much, Gasper. And for all of the participants, thanks for joining from all over the world. And have a nice day, everybody.